Hi, everyone, and welcome to episode 601 of People Behind the Science. I'm your host, Dr. Marie McNeely, and today we are going to be chatting with our guest, Dr. Amanda Therrien. And listeners, if you want to continue meeting more exceptional scientists through our show, you can follow us on Twitter at PBT Science, and I'm at Ph.D. Marie. Today, Amanda is going to share some wonderful stories and insights from her life, her research, and her career. So get ready to meet another one of our brilliant people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am delighted to be speaking with our guest scientist, Dr. Amanda Therrien. So, Amanda, welcome to our show today. How are you?
I'm doing well. How are you doing?
I'm doing quite well, and I'm excited to chat more with you about your life and your research. But let me first start by giving our listeners a little teaser here, a little bit about your background. So, listeners, Amanda is an institute scientist and director of the Sensorimotor Learning Laboratory at Moss Rehabilitation Research Institute, or MRRI. She received her bachelor's degree in Human Kinetics from the University of Ottawa and her Ph.D. in Kinesiology, specializing in sensorimotor neuroscience, from McMaster University. Before accepting her current position at MRRI, Amanda conducted postdoctoral research at the Krieger Institute and at Johns Hopkins School of Medicine. And she is here with us today, listeners, to talk more about her science and her life. So, Amanda, we want to get to know both sides of you today. So can you start by telling us what you like to do when you're not doing science?
Oh boy, I feel like I like to do a lot of things. I feel like my interests are quite eclectic. As you said, my background is in kinesiology, so I do really like a lot of activities that just kind of get me moving, get me outside. I'm a pretty avid runner. It's often how I blow off steam, and anytime I travel, I like to explore through running. But I also like kind of more quieter activities. I am a really avid knitter. I really like gardening. I read a lot. I like cooking. I'm outside of running. I feel sometimes like I'm a 90-year-old woman in a 30-something-year-old body.
Well, I've tried my hand at knitting and made a couple of really bad-looking scarves. What is your specialty in the knitting world?
Well, I like all kinds of knitting. I feel like I started a little bit to offset some of my guilt for watching too much TV. I felt like, if I can make a hat, it makes up for the fact that maybe I binge watched an entire season of The Office. But I really like it. I think as a scientist, so much of what we do is playing kind of such a long game that it's nice to have a hobby where you produce something in a relatively short period of time and the stakes are kind of low on it. So just sort of a repetitive activity. I can produce things that are useful, and it has a nice design element too. I quite like textile design, so it ticks many boxes for me.
Fantastic. Well, and you mentioned you're an avid reader too, and we love talking books on our show. So we'll jump right to that. Do you have a particular book that you've read recently, or maybe just an old favorite that you want to recommend to me and our listeners?
When I think about books that have had a big influence in my life and that I've read a couple of times or more than a couple of times, one science book that I love and recommend, particularly for perhaps younger folks who may be listening to this, it's a book called Wonderful Life by Stephen Jay Gould. It's about the Burgess Shale, which for anyone who's not familiar with is a really big fossil record that exists. And I first read it when I was in high school after my biology teacher recommended it. And it was really kind of the first science-y book I think I had ever read for pleasure. And it's just a really wonderful book. It really made me love science and biology in particular. So I highly recommend that one. I guess a non-science book to go with it would be one of my favorite books that I've ever read. I've come back to it many times over the last 10 years, especially. It's called Conversations with Myself by Nelson Mandela. It's really a book that's just a collection of his writing. So journal entries, letters, etc., that span a big chunk of his life. It's just this sort of very unedited collection of letters. And it's a really lovely book for many reasons, but I think it came out when I was in my early 20s, and I think I was at a developmental stage where what struck me most about it is how much you can see his thinking change over the course of his life. And that's sort of a lesson, I think, that has stuck with me a lot, both personally and professionally in my science, is that never get too stuck in a certain mindset, because your thinking is likely going to change as you get more information.
Oh, definitely. Well, excellent recommendations, Amanda. I look forward to adding Wonderful Life and Conversations with Myself to our list on our website for our listeners out there. And we've touched on the science just a little bit, but I'd like to get into a little bit more detail here about what you're working on now. So how do you describe your work to someone who's maybe not in science at all or not specifically in your field?
So I'm a researcher in the field, as you said, of sensorimotor neuroscience. So really, this means that I study how your brain integrates sensory information that comes in from the body. So things like vision, proprioception, which just describes kind of how your movements or how you know where your body is in space when you can't see it, and how that information is integrated with commands that are sent from your brain back down to the body to control your movement. The way I do this primarily is I study human behavior. And what we can do is by precisely measuring how individuals, both neurologically healthy individuals as well as individuals with neurological damage, make movements in different situations where we alter the sensory information they have available to them. So, for example, by manipulating visual information that they have about their movements, or by using a robotic device to alter how a movement feels, we can gain quite a lot of insight into how the nervous system works, as well as how damage to different brain areas can disrupt movement control.
Very interesting. Well, Amanda, I'm excited to talk more about specific projects as we go through our conversation today, but I'd love to talk a little bit about motivation. And I know we tapped on some wonderfully motivating people in talking about books already, but do you have a favorite quote or a saying or just something that really motivates you in your everyday life?
Yeah, it's funny. I will admit I listened to some of the previous episodes of this podcast, and I know a lot of scientists quote George Box, the famous quote of all models are wrong, but some are useful, which is very true. And I think it looms large in a lot of scientists' lives. But I suppose in the interest of variety, another one that I really like is from Albert Einstein, and it's something along the lines of, I don't remember the exact wording, but basically that you can't solve a problem with the same mindset that created it. And it really inspires me to think outside the box, which I think has really benefited a lot of my scientific pursuits.
Oh, definitely. And we touched on just some of the different steps that went into getting to your current position today. You know, as a scientist, you get to work with a lot of cool people and you get to meet a lot of amazing, inspirational people throughout your life. So, Amanda, when you think back, are there particular people that really rise to the top of your mind as mentors or role models or maybe historical figures that you looked up to?
I've been really lucky. I've met so many wonderful people who I admire a lot. Particularly as a scientist, I've been really lucky to have worked with some pretty amazing women who are really just kind of badasses. My postdoc advisor, Amy Bastian, was someone whose work I had first encountered actually as an undergraduate student and was a big factor in why I pursued science as a career. So the fact that I ended up working with her for quite a long time was pretty amazing, and I'm pretty grateful for that experience. She taught me so much about science, but not only just as a role model for how to be a woman in this career. One thing that she really teaches all of her female trainees is to really be confident and don't apologize for your seat at the table. Take that seat and run with it. And I'm really grateful for that. One of my other women, there's so many that I really admire, Nora Fritz, who's one of my best friends, and she's a faculty at Wayne State University in Detroit, is just wonderful and so strong. And now at MRRI, I work with Laurel Buxbaum, who's another incredibly strong and amazing female scientist that I really look up to.
Oh, phenomenal. It sounds like you've had some great people to serve as these role models and mentors. And I'm glad you mentioned Amy. She's actually someone that I've looked up to and really admired as well with my background in Parkinson's research and movement science. So what was it like for you maybe stepping into her lab for the first time as a postdoc and just kind of jumping in there?
I feel like by the time you start a postdoc, you've interacted with somebody so much that some of the surrealness of it had worn off by then. But I've always felt very lucky. It's a really great group that she runs, and the lab itself is a couple of PIs and they all kind of share the lab space. And it was just a really fun group to work with for the 6 years that I was there. And You work really hard, but it's a good group. And she's a really pretty amazing mentor. She is very motivating. She cares about her students. She cares about their well-being. And she's a very, very good teacher of how to design experiments and how to conduct kind of research that has, a big thing for me was how to conduct research that has both basic science and an eye for clinical rehabilitation practice.
Phenomenal. Well, Amanda, I'd love to rewind back even further than before your postdoc days. Can you give us some insight into where your interest in science started? You mentioned that already in high school, you sort of had this interest in biology perhaps blooming. Where did it begin for you?
I never really thought that I would be a scientist. No one in my family is. I had no concept of kind of what that looked like. When I finished my undergraduate degree, I had done a part of the honors project. I had chosen to do a research thesis. So I did a stint in motor control lab in the kin department at U of O. And I really liked it. And funny enough, it was a project looking at cerebellar ataxia, which is sort of a big focus of my current work today. But what's sort of strange is even throughout that experience, I hadn't really thought that that would be the career path I would go down. I feel a little bit when I think back about my academic career that it never really feels like a straight shot. It was sort of this series of decisions that were just kind of made. It didn't feel particularly impactful at the time, but I was finishing my undergraduate degree. It was 2008, so there was a recession starting. And I had originally thought I would go to medical school or teacher's college. And in Ontario at the time, for some other reasons, both of those professional programs were seriously limiting how many admissions they were taking. And so I started to doubt whether that was the best choice I could make at that time. And my honors thesis advisor was moving from U of O to McMaster University. And an opportunity arose for me to do a master's degree with him. And I really liked the project and I thought I'm pretty inspired and I would like to learn more. So I just kind of went with it. And a year into my master's degree, McMaster had an option. You could just transfer directly into a Ph.D. program.
Oh, wow.
If you went through this sort of special defense thing instead of finishing your master's degree. And again, it was just like, I really like what I'm doing. I'd like to keep doing it.
Right. Going well so far. Yeah.
So I kind of did that. And by the end of my PhD, I was really lucky in both my doctoral degree and in my postdoctoral training that I was given the freedom to really kind of pursue ideas that I was very interested in. And I say lucky because I know that that's not always the case for everyone. A lot of it depends on grants that a student or trainee may be funded through and how restrictive the rules are about that grant. And I was lucky that both my doctoral advisors and my postdoctoral advisors had the flexibility available to just kind of, and then they were open-minded to sort of just letting a research program go where it was going to go. So by the end of my PhD, I again was in this state. I thought, I really like this. I have so many more questions. And I thought back to Amy Bastian's work. And it's kind of crazy to think about now because I really just cold called her. I had never met her.
Wow.
I cold emailed, I suppose, rather. And I just sent her an email saying, I'm Amanda. I'm finishing my PhD at Mac. And here's my CV. And here's kind of what I'm interested in doing. And do you have an opening in your lab? And then I didn't hear anything for a couple of months. So I just thought, well, whatever. And a couple of months later, she emailed back and we started chatting and realized there was a project that we were both interested in. And it just progressed from there. So it feels like another series of decisions. I've just been really lucky that I was able to pursue ideas that I was interested in and really kind of passionate about.
Certainly. So in that couple of month interlude before she replied, had you been looking into other opportunities or trying to make plan B?
I feel like this is where perhaps younger listeners don't emulate my behavior because I didn't really, thinking back now, I don't really know what I had in mind at the time. I think I just was, I'll figure it out when I finish my PhD, which is incredibly naive and not something I totally recommend. But yeah, it's sort of, I think, what feels strange and it gives me a sense of gratitude that it did in the end kind of work out.
Then how did you find your way to MRRI?
Again, kind of the same thing. I was finishing up my postdoc and I was looking for jobs and saw the posting for position with the movement science discipline at MRRI and I applied and everything clicked.
Oh, wonderful. And you've got some awesome projects starting in your new lab there. So is there one in particular that you want to tell us more about?
Yeah, so one line of research in the lab is trying to understand whether we can optimize therapeutic interventions for a movement disorder called ataxia. Ataxia is caused by damage to a brain structure called the cerebellum, and it's marked by impaired movement coordination. So the cerebellum, we think, is integral for a process called sensory prediction. The nervous system, or our motor control system, needs sensory prediction because, in truth, the processing of sensory feedback from your body is quite slow. So, for example, everything you're seeing right now and listening to this podcast, everything you're hearing right now, actually happened in the past. Depending on your nervous system, it happened about 100 to 120 milliseconds ago. So that may not seem like a very long time, but it's quite problematic for movement control. So in the case of ataxia, where we think sensory prediction is impaired, the nervous system has to rely instead on time-delayed sensory information. And this forces movement control to use feedback, this delayed sensory feedback. And what you get is this kind of negative feedback control mechanism. So this causes oscillating behavior, which is what we see with what we call ataxic tremor. So sensory prediction is not only crucial for movement control, it's also integral for an important mechanism for learning new movements called adaptation. Adaptation uses information about the direction and magnitude of your movement errors to keep those sensory predictions up to date. And this process is necessary because we live in a world that's constantly changing. Many of your listeners have probably experienced sitting down to a new computer and it takes a minute to adjust to how the movement of the mouse produces movement of a cursor on the screen. Or you get into a new car and you have to adjust to how much force you need to apply to the gas pedal to accelerate the vehicle. So what we're investigating is whether a novel intervention that really strives to remove this kind of problematic vector error information, so this particular direction and magnitude of movement error information, whether this can improve training outcomes for this disorder. So to give you an idea kind of how we do this, with arm movements at least, we can do this by removing visual information of the arm during movement. And we can do this with virtual reality kind of environments. And what we do is instead of giving them direction and magnitude information about their movement errors, we just give them binary signals. And what we're trying to do with this intervention is leverage a mechanism of learning that is less dependent on the cerebellum. And the important thing here, what we're trying to do is we're not going to learn someone out of cerebellar damage, but what we can do is teach them a way to achieve a functional outcome. So in a very simplistic case, moving their hand to a desired endpoint location in a manner that is smooth and direct by leveraging a training mechanism that is less dependent on the cerebellum.
Oh, very interesting. And you mentioned specifically looking at arm movements. Are these particularly impaired with people with cerebellar ataxia, or is all of their movement impaired?
We're starting with arm movements, but ataxia affects all the body effectors. So everything from balance control and then by extension walking, as well as eye movements and speech. What's useful to us about starting with arm movements is that we kind of know, or we feel reasonably secure in thinking that visual information is the dominant source of the direction and magnitude of movement errors. There's a lot of research even in infants to suggest that as humans are just with upper extremity control are just very visually dominant. One thing that's interesting about this line of research that I think will be things that we look into moving forward are whether these kinds of interventions can be used for other movements. And this is going to be a bit tricky, especially in cases such as balance control, where sussing out that dominant source of vector information and determining whether we can manipulate it is maybe not as straightforward.
Oh, wonderful. Amanda, this is a fascinating project. And I know myself from personal experience that working in clinical populations often comes with its own challenges. And I think as a scientist in general, you face a lot of difficulties, struggles, and challenges on a day-to-day basis. And I think it's so important to talk about these and share them with our listeners who might be in the same boat themselves. So when you think about your own career and your own experiences, Have you had a major failure or a particular struggle that was particularly memorable that you can tell us about and maybe explain how you worked through it?
One that honestly comes to mind is, as you mentioned, I've only been at MRRI for a little over a year now. And this past year has honestly been quite a struggle.
It has been unprecedented.
Yeah. I started my current position a little less than 4 months before the pandemic hit. And it's been an interesting challenge. Navigating the transition from postdoctoral trainee to independent investigator is a challenge and is intimidating, I think, even at the best of times. And then you throw in kind of logistical challenges of COVID into the pool, and it's tough. It's tough both professionally and personally to have something so vastly change from what you thought possible or had any kind of expectation of. But in terms of how to get through it, when times are tough, you try to keep your eye on the prize a little bit and think about why you're doing this. At the end of the day, I really like what I study. I feel very passionate about it. And that just gives you a little bit of a push to just take it one day at a time and try to focus on pushing that boulder just a little bit. And some days you're going to push the boulder more, and some days you're going to move backwards, and that's okay. Because like I said before, we're playing a long game here, and you got to try your best to think about how far you've actually pushed the boulder. If you took an aerial view of the situation, you've actually moved the boulder overall. So
definitely. And I can't imagine trying to really launch a new research lab in a pandemic. So what were some of the biggest maybe challenges or logistical issues or problems that you encountered trying to navigate this?
Logistics are a funny one. I mean, I'm sure everyone remembers, even if you're past this stage, you remember the transition from trainee to independent investigator, and others may be going through this right now or thinking about it. There's so many behind the scenes things about running a lab that as a trainee, you never experience. So learning about those while simultaneously learning about how the normal procedures of handling those logistics are now no longer applicable is an interesting experience. Early career scientists, it's a kind of tough moment in your career for productivity. You want to be productive. You want to be putting in grant applications, and it's a challenge to do that when it's quite difficult, for example, to collect pilot data for grants. And personally, it took me a while to really let myself accept the more sort of personal side of it, which is for so long, you're working towards this thing that is kind of a goalpost in your mind of one day I'm going to have my own lab. And it's hard not to put a lot of emotional weight, I think, on that. And then to get there and you're so excited, and then to very quickly kind of have everything change in so many unexpected ways, I think has also been a challenge.
Sure. Well, thank you for this candid description of some of the difficulties that you went through. I think it is so important, like I said, for people out there who might be going through the struggle themselves to know they're not alone. But we don't want to just talk about the difficult times. Amanda, I'd love to talk about your successes. So do you have a favorite success, whether it was a big win or just a small but particularly meaningful one, that you want to share with us today?
The big wins are fun. You can think of the big wins like a big paper comes out or you get funding. And those are wonderful, but they're less frequent than I think we'd all sort of like. I really like the small wins. And I think one recently that has been encouraging is that I think one thing when you're starting a new lab and the learning curve is quite steep, it's a lot and you kind of lose your mojo. I kind of lost my mojo a little bit for a while, and that was a little bit tough. And I think recently I've noticed that the creative juices are kind of starting to flow again. And I have some new collaborations that are developing, and we have some really exciting lines of research in the pipeline. And to me, that's my favorite part of this job. I really like just that process of problem solving and designing a new research program. And it's been encouraging to kind of have that going again.
Absolutely. So what has this, I guess, transition to things maybe working a little bit better been for you? Has it been slow and steady, or was it sort of like a switch clicked overnight and you were like, wait, I've got some great ideas. We're going to start moving these projects forward? I
think kind of both. I don't have switch clicking very often. Mm-hmm. I feel like maybe for me, ideas tend to percolate, and perhaps I'm not alone in this. You can be a little bit impatient about they're not percolating fast enough. Right. But once you've got your metaphorical pot of coffee ready, it's quite rewarding and exciting. So I would say probably a little bit of both, but mostly the slow trickle.
Certainly. So is there a particular new line of research or collaboration that you are especially excited about?
Yeah, actually. So Aaron Wong, who's another PI at MRRI, who I knew from back at Hopkins, he and I are starting a really exciting line of work looking at the sense of proprioception, which is the sense of how your movements feel and kind of where your body is in space without vision. And there's a lot of work that's been done on what we might call a static sense of proprioception. So how do you know where your limbs are in space when those limbs are not moving? And one thing that he and I have had a lot of questions about over the years is we study motor learning. And a big part of that is you need to have these very rapid sense of where are your limbs in space while they're moving. And sort of the processing of that information is a little bit different than what happens when the limb is in kind of a fixed position. So we're starting some new studies that are going to tease that apart a little bit.
Oh, very cool. Well, good luck to you on these new studies you're launching. And this next question has been, I think, particularly painful lately because this is one of the sad things about the pandemic is you haven't been able to travel as much for science and haven't been able to kind of see those colleagues and collaborators in person. But when you think about all the places your science has taken you thus far, do you have a particular place or a conference that you went to that really sticks out in your mind as the most impactful or most memorable?
Oh, yeah, that is sad.
It's been a while, right?
We can't travel anymore. I don't think that the lack of travel, I think in some ways it's opened people's eyes a little bit to some flexibility with conferences that are potential benefits for accessibility as well. But yeah, traveling, there's one that sticks out, I think, for a couple of reasons. In 2019, I had the opportunity to co-chair one of the pre-meetings for a Gordon Research Conference on the cerebellum. In this particular, in 2019, the conference was in Les Diables in Switzerland, this town out of a storybook in the middle of the Swiss Alps. Mm-hmm. So it was already just sort of this surreal experience because you look out the window in the morning and you think you're in sort of a Hans Christian Andersen story. But Gordon conferences as well are just, for anyone who's not familiar with them, it's this sort of private organization in the United States, and they run many different conferences on many different topics, but there are these really small meetings that are very focused on a very specific topic, and they're kind of intense, but they're usually about a week long, and everybody is sort of in this spot and you're sort of together all the time. You stay all in the same place, you eat meals together, and you go to these talks. And the nice thing about the data that's presented at these meetings as well is the kind of rule is that it's very new data. So it's not published. And in many cases, the data collection process is ongoing. And so the small group being together all the time and these discussions of these new ideas that everyone's really excited about make these conferences really fun. And Gordon meetings are ones that I always come back and I would feel very inspired and refreshed and kind of ready to get back into work. So going to that conference in the middle of this storybook setting was this sort of very surreal experience of like, this is sort of the fairy tale imagination of what it's like to do science.
Oh, that's wonderful. So were you able to spend a little bit of time sightseeing there in Switzerland as well while you were there?
Yes, thankfully. And I don't ever do this. It was the only time I've ever done it, but I'm so glad I did. Mm-hmm. So beforehand, a friend of mine had invited me to give a talk in Lausanne. So I went there for a couple of days and visited with him. And then after the Gordon meeting, I went to visit another friend who lived in Zurich. And then after that, I was able to fly to Belgium to visit two of my friends that live there. So it turned into this really wonderful opportunity to catch up with friends who are otherwise very far away.
Oh, that sounds phenomenal. So in terms of, I guess, destinations or sites to see, do you have any particular recommendations once me and our listeners start traveling again?
Yeah. Aspirational.
Yeah.
I mean, go to the Swiss Alps, honestly, it's unreal. But one thing that was really fun and really unexpected in Zurich, there's the Limmat River that runs through the city, and it's incredibly clean. And a big thing to do in the city is apparently you just go down to the river and you bring flotation devices and you just float down the river, but it's in the middle of the city and there's like pubs and bars like right up next to it. So you can like get a drink and they'll give it to you. And then you just go get back on your floaty and you go down the river.
Oh, wow.
And it's this kind of crazy experience because it's sort of this thing that river floats, I always associate with like some off the beaten path kind of thing. And it's right in the middle of Zurich.
Mm-hmm.
And it was really wonderful. It was just a really great time.
Oh, that sounds amazing. Yeah, every float trip I've been on is sort of way far away from civilization. So this sounds very novel.
Yeah, it was so strange. My friend was the whole time, she was just sort of laughing at me because I was like, this is incredible.
Well, it sounds like an amazing experience. And I think, as you sort of hinted at, the people you get to meet and work with as a scientist help make the career so wonderful. And I think this network of people you develop, you often experience quirky traditions or just see a different side of them that I think the public doesn't always get to see. And I think sharing these stories is really important because scientists are often portrayed as being very serious, very critical, and fitting a certain stereotype often in the media. So, Amanda, do you have a story that maybe showcases this more human and fun side of science that you can tell us about today?
You're totally right. It is this sort of funny thing, especially I feel like I remember back to being a student and these people who you read their papers and they almost seem kind of not real to you after a while. And then you meet them and it's really wonderful. It is a really wonderful thing. One thing that always makes me smile is I think back to the lab that I postdoced in. As I mentioned, Amy Bastian's one of the PIs, but there are a bunch of others. And when I started, it was 3 PIs. I think now that group is up to 5 or 6. So it's a big group of people. Pablo Selnick is another PI with that group. Kathy Zakowski was there for a while. Vikram Chib is there as well now, and Ryan Remick. And one tradition that we had that I loved, we called it Park Day. It was one day, usually in the spring, early summer, before temperatures regularly start to exceed 100. And everybody takes the day off work. We all meet in a park and we just play games, play volleyball, play can jam, have a little barbecue lunch, and just let loose. And one thing we'd always do, part of Park Day, was, I don't know if you can call it, it's like an obstacle course combined with like kind of a Tough Mudder race. But it was this fun competition that we designed this super crazy kind of race, and we'd break off into teams and compete for fun. And there's a picture out there somewhere. I don't know who has it. I'd like to get my hands on it because the memory of it makes me laugh, but it's a picture of Amy Bastian just tackling me at the waist, like as part of this race. And they were always just great days to bond as a group and blow off steam from work. And it's something that I would really like to start with my lab at MRRI as well.
Oh, wonderful. I think studying the cerebellum and sensorimotor neuroscience, you got to work in the physical activity and movement in there somehow. So Park Day sounds amazing.
Yeah, exactly. It's all for science, really.
Of course. Appreciating a different side of your work.
Exactly.
Well, you are answering some phenomenal questions in the lab. And I know we talked about some of the challenges that scientists face. And I think often things like funding, staff, technology, feasibility, time, pandemics, whatever the case may be, stand between you and being able to answer your dream research questions. So, Amanda, I want you to dream big with us today. If we took away everything that normally stands in your way, what is the one question you want to answer most?
There's one question that I would really love to answer, and I think a lot of people would love to answer this, is I would love to start doing a real deep dive on hemiparesis, which is a condition that results generally from stroke affecting the corticospinal tract. And while it is called one thing, and it presents, I should say, clinically as if you see someone who has a stroke on one side of their body, they show weakness and spasticity in some muscles. And hemiparesis is a bucket condition that broadly describes that. But in reality, hemiparesis is so many different things. And I think it's something that we don't really understand fundamentally what all these factors are. So one particular question that's very interesting to me is there's some work that came out a couple of years ago from a friend of mine named Rob Hardwick, who, when he was working with Pablo Selnick and John Krakauer at Hopkins, that there's this interesting question about stroke and about kind of motor rehabilitation, which is if you compare stroke, for example, to cerebellar ataxia, with ataxic individuals, if you ask them to make a reaching movement and you ask them to do, 100 repetitions of a movement, some percentage of those movements are what you might think of as quote unquote normal looking. And that's fascinating to me because that suggests that the nervous system is capable of producing relatively coordinated movement. The question there is, why doesn't it do that the majority of the time? With stroke, I think what's not clear is how much of that is true when you have corticospinal tract damage. In some cases, it might be. But one thing with corticospinal tract damage is that now you have made some movements not possible. And so I think an interesting rehab question is, can we figure out what movements are possible and see if we can kind of move around that space? And work by my friend Rob Hardwick has shown that if you can figure out this envelope in which someone with a stroke can change their behavior, they can change their behavior within that envelope. And perhaps this raises questions about some of the thinking around rehabilitating individuals who've had a stroke. Is perhaps we need to be a bit careful about identifying the set of movements that perhaps are no longer possible and try to gear the rehab intervention towards the set of movements that are still possible. So when I think pie in the sky kind of ideas about that, one thing that comes up for me is this idea called feasible set theory. And it was developed by a guy called Francisco Valero Cuevas at USC. And it basically states that if you could start with a musculoskeletal model of a limb, you could figure out all the possible movements that that limb can execute. And you can kind of map that landscape. And one thing I wonder about with stroke is, with that as a framework, could we look at this complex landscape and could we start to sort of see how corticospinal tract damage in different ways changes that landscape, that feasible set? And once we have that feasible set for an individual, could we start to look at how people kind of move within that feasible set? Can we identify an optimal movement and try to bias them towards that within that sort of envelope or that region of the landscape that's still available? And why I think it's so that pie in the sky perhaps is Francisco Valero Cuevas's formulation of this model is so many years of work, and it was just to get the thumb.
Wow.
That musculoskeletal modeling part is no joke. Mm-hmm. And you try to do that for the arm, where you have many more joints, and the shoulder joint is a ball and socket joint. And computationally, it would require massive collaboration and many, 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 many years of work. But it's something I think about.
Oh, well, Amanda, this is fascinating. I appreciate you sharing these ideas and giving me and our listeners something to think about today. And I know we've covered a lot of ground in our interview thus far. I'd love to wrap up our conversation talking about advice. So when you think back about your own experiences and perhaps your career, did you ever receive one piece of advice that you think was really helpful that you want to pass on to our listeners?
I think a lot about this thing that Amy used to say, or she still says all the time, which is the data is the data. And what she means by that is, as scientists, we spend so much time reading and finding a research question that we think is meaningful. And then from that research question, developing a hypothesis that we think is reasonable and makes sense. And then from that hypothesis, we spend a lot of time designing an experiment that we think is a valid way to test that hypothesis. So when you reach the stage of data collection, it's tempting and it's very hard to resist. Again, attaching kind of emotional weight to that data and attaching a bit of perhaps subjectivity to the data. And one thing that Amy stresses a lot, which is try to just remain almost agnostic at that stage. You've done your due diligence, you've designed the best experiment you can, and let's see what happens. And don't be afraid if the result is unexpected. And indeed, I can tell you that this is the case with my career, and I'm sure it's the case with many scientists that you'll speak with, that the unexpected results are often the ones that end up being the most interesting and yield the most fruitful programs. And so perhaps in some ways it's not unrelated to the lesson I took out of conversations with myself, the book, that try to be open and stay curious and not to have too many set expectations about what your experiments are going to show.
Oh, definitely. I think it's, like you sort of alluded to, so difficult, but so important to kind of stay open-minded and not get too disappointed if things don't turn out as expected.
Yes, because they won't a lot.
Of course. Well, wonderful advice for our listeners. Amanda, is there any last note that you'd like to end on, or a last piece of inspiration that you'd like to share with everybody today?
Career-wise, I'm new in my role, But I guess when I think back on my career thus far, I feel very grateful and I feel lucky that I was able to do this. But I really think it's good to study something that you fundamentally really like. By that, I mean, as you move forward in an academic career, sometimes you're going to encounter a point at which the question you want to investigate and perhaps what is a bit more sexy or more attractive to a funding body is something different. And it can be tempting to kind of go in the direction of what is perhaps more popular. And it's totally understandable. But as we talked about earlier, there are times in this career where it can be really tough. And I think the thing that will keep you going through those tough times is if you're really passionate about what you do and there's a reason why you want to study it, that'll keep you going. So I guess I'll borrow from Tina Fey and say, just do your thing, do it well, and don't worry so much about what other people think.
Oh, I love it. Well, wonderful message to share with all of our listeners out there. If they are intrigued and they want to learn more about you and your work, Amanda, what is the best way for them to do so?
I am on Twitter. My handle is at Amanda S. Therrien. I will admit I don't tweet very often, but you can find me there. The other spot would be most likely to check out my lab webpage, and you can get links to publications through the MRRI website at mrri.org.
Perfect. Well, listeners, definitely follow Amanda on Twitter. Check out that website to get more information. And Amanda, it's been such a pleasure to chat with you today. Thank you.
Thank you.
And listeners, great to have you here with us as well. We'll see you next time on another episode of People Behind the Science.